Hello, everyone. I am recording this so that if there's any connectivity issues with our presentation, you've got a backup. This is covering a more in-depth discussion in regard to the BiomeFX. We went over the introductory, covered the surface of why it's different from other stool tests, but let's go, into the weeds and really get some good questions out of what we're trying to achieve here. Okay, so the objective of the introduction presentation was to let you know about our partnership with Cosmos ID, why we went into the stool testing business, and my personal story. I was a practitioner. I had done tests, quite a few on clients as well as myself, dating back, got some history there. But it's still, this is really kind of a pharmaceutical approach. We're now as technology is getting better, we really have to look at stool testing, as a functional approach. It's not, the pharmaceutical approach is, you're doing something to make a huge change, but many times there's a side effect to making that change. If it's stopping enzyme productions or stopping certain biochemistry of the body to elicit an effect, then there will be side effects. In the stool testing arena, if you're looking, in a narrow scope and just seeing a pathogen and then waging war on that pathogen, you're not taking into account, is that pathogen being handled by the microbiome with no problem, and will waging a war, cause more problems? So this is where we're at in the stool testing, is now being able to see more of what's going on, and we can come up with better strategies because we're understanding more how the microbiome, is functioning. Okay. And this is an important point to make as well. The credible universities, research facilities, and agencies out there are using Cosmos ID. They're not using Genova, Diagnostic, and some of the other tests that are out there, because they're not accurate enough. So that's very telling. Now, what I'm hoping to, cover here is really get into some of the interpretive concepts, how to interpret the result report, and really, introduce you to the power of the potential of this test as it's evolving, how the phases are being introduced, and what you'll be able to do with that, and go over some case studies, help you connect the dots and really drive it home. Right, these videos are available to you to go through. They cover, the more times you hear this information, every time you hear it, you'll glean new information. And when you hear it from different people, again, you'll get different perspectives. This is Kieran in these videos explaining, why we're doing this stool test, why we partnered with Cosmos ID, who Rita Caldwell is. Colwell, she, you'll see her bio there, very, very credentialed, 800 different publications. She will be the keynote speaker at the keynote microbiome meeting in Scottsdale, Arizona. Another point about Cosmos ID is that because they were the lab that did the global microbiome test and the American gut test, they have the biggest reference of samples, and that's very important. what samples you're referencing to make all the difference. Right. So yes, we can give you the 30,000-foot view and give you a better idea of what's going on. If you know what bacterial ratios, you know what to do, then you can deduce what is going on in the gut. But the problem with the other labs is just the accuracy of really identifying those bacteria. So when they made ratio comparisons, they wouldn't be accurate either. Okay, and this, Kieran goes over just the basic limitations of any stool test, is that you're still limited to the colon and the distal colon. But because we have a more advanced sample technique, because we use a coring tool, you're going to get better representation. But it's still what it is. All right. Methodology of 16S ribosomal RNA versus the whole metagenomic DNA sequencing. And there's some great resources to some independent studies that discuss, this dilemma of, getting better results because you're looking at the whole genome. He discusses how long, it takes to see changes. Sample report section by section. And we're going to go over some of the interpretation concepts on those. Once you get your sample report, we have experts that are on call to set up an appointment with to go over those reports before you discuss them with your patient. There is a printed interpretation guide, and then this covers the previous slide of the practitioner training videos. Okay, so the first section to discuss is the diversity. And you've seen the sample report. We know the alpha diversity is talking about richness, how many different spores. The more diverse the organisms, the more diverse the microbiome, the metabolome or microbiome, or the metabolic metabolites. Absolute number of species, quantity, interstate, whatever way you can conceptualize this to explain it, you'll develop your own technique. I like to have certain questions like this, when you're saying alpha diversity is how many species inhabit your gut, where beta diversity is do the right species inhabit your gut. So that's, conceptually, good questions when you're explaining the difference between the two. Because people may not relate to, richness versus composition, or, quantity versus quality, or interstate versus intrastate. That may not resonate with them. So it's a matter of finding your own description. If you have high alpha, versus low beta, it's saying that you've got, protective organisms, but it's not protective in the environment that you're in. And that's what we're looking for. And it means that you have many different species, but they are different species from the US population. And then you want to ask yourself, why would that be? Has this person been traveling or living abroad for a significant amount of time? Does this person eat radically different than the standard American diet? So there's, you're going to have to explore the reason for that. The only way you can have accurate reporting is to have representative global library of samples. And that's what I was referring to before. The only Cosmos ID achieves this because it was the lab that was used for the microbiome project and the American Gut Project. And the gut microbial populations adapt to the environment, ratios change, levels of seeded keystones are influenced, pathogens are encouraged or discouraged by the terrain. And then, there's a whole science of studying microbiome diversity. And what this is talking about is these eigenvectors are what you see in that test report. and they are basically statistical latitude and longitude lines for each species. So just know that there's a lot of background behind the science of this test. Now, the resistome, another section of the test, is really discussing economics. So, are the organisms producing metabolites that help their neighbors or hurt their neighbors? Are they thriving or being suppressed? Are they getting along with each other? So it's a supply and demand environment, and there's competition going on. If there is not enough competition, then the ecology is off. Somebody is trying to be a dictator instead of, it being run like a democracy. So a healthy microbiome has healthy boundaries. It talks about stability and uniformity of a society that's dependent on the individual freedom of expression. It's kind of a microcosm of, our human society. Bacterial equilibrium is their expression of their Bill of Rights or their equality. Balance or homeostasis. Imbalance will be reflected in the alpha and the beta diversity. A rich species mix, well adapted to the environment, will have a high resistance or resistome, or be resilient to infection. The pathobiome. Presence or occurrence, not always an indication of dysfunction. And this is why other tests can lead you astray. Just because it's there doesn't mean it's causing a problem. You got to look at abundance, not always an indication of dysfunction, without taking into context the healthy bacteria ratios. Effectivity or strength, how effective or strong is your microbiome at dealing with pathogens? And then finally, the control index. Higher the better, indicates that the pathogens are being dealt with well or being controlled by the commensal organisms. So higher the number of pathogens that are being dealt with well is a sign of a healthy microbiome. There is, if you look at the other studies, there's no studies that target specific pathogens. So just because they tell you one's there, they have not looked at those killing protocols to see if they're even effective. Okay, the dysbiosis ratios. We're talking about the Firmicutes in ratio to the Bacteroides. A high Firmicutes to Bacteroides is usually indicative of obesity. Low is indicative of reduced inflammation. The ratio of Proteobacteria to Actinobacteria, high proteo is going to be a poor metabolism. Low is going to be a healthier metabolism. The Proventella versus the Bacteroides, high is generally a low body mass index and reduced inflammation, carbohydrate and fiber consumption. A low is generally obesity and high protein and fat consumption. Now, in the biologically active bacteria, we're looking at ammonia producers, sulfate producers, and methane. But what's just as important as, do you have bacteria that produce these gases? Are you able to remove them? What is the body's ability to deal with it? So a high level ammonia reducing bacteria converts more protein into ammonia, which stresses the liver. So increase the pH, and it increases the pH of your gut, more susceptibility to pathogen occurrence, fungal overgrowth, type of dysbiosis that leads to leaky gut. The sulfate, if you have high levels of sulfate reducing bacteria, it means foods that tend to be high in sulfates, do not clear well, will cause problems. You're going to convert more of that sulfate from these foods into hydrogen sulfide, which then becomes toxic and inflammatory to your bowels. Methane, people that tend to have low sulfate reducing bacteria tend to have high methanogens because they're producing less hydrogen sulfide. They're producing more methane from the metabolization of food. The methanogens produce high levels of methane, These people will have gas response from their food rather than inflammatory response. The estrobolome. There are 60 types of bacteria that are associated with high beta-glucuronidase activity. So this enzyme conjugates estrogen into its active form. Increasing activity is associated with high fat, high protein diets, increasing circulating levels of estrogen. Alternatively, high fiber diets are associated with decreased beta-glucuronidase activity. So if you deal with hormones, you're balancing hormones, you're using detoxification protocols for hormones, you're giving bioidentical hormones, then you really have to be well-versed in how the estrobolome is functioning. You need to know if your client is going to be able to tolerate these protocols you're putting them on based on their gut bacteria and ability to handle estrogen. So, and this is going to give you a good indication. Another section of the report deals with functional keystone strains. And these 8 are particularly the ones that we focus on. The 3, the Akkermansia musciphylla, the Faecalibacterium, and the Bifidobacteria species are the ones that we have focused on for the Mega Mucosa product. But you see there are other keystone strains, and they play key roles. Akkermansia musciphylla is helping the metabolism of the short chain, increasing short chain fatty acids. The Rhuminococcus bromai is a cellulose degrader and involved in skin health. Faecalibacterium perniciae is really the one decreasing inflammation and increasing short chain fatty acids. Rhuminococcus flavinifaciens is a cellulose degrader. Roseburia intestinalis is involved with the metabolization of beta-mannan degrader and a butyrate producer. The Eubacterium rectile is a butyrate producer. The Bifidobacterium longum is an acetate producer, and Lactobacillus species also is a lactate producer. All right, and as promised, we're going to talk about some of the new additions of the BiomeFX Phase 2. One of the additions will be saccharolytic fermentation. This is going to be very interesting. So we know that high levels of saccharolytic fermentation are associated with high metabolism and reducing intestinal pH, which supports a healthy gut environment. Sac fermentation levels may be low as a result of high protein diet, a low fiber diet, or insufficient keystone species. The relevant bacteria that we would be monitoring is the Rhuminococcus bromai, the Lactobacillus, and the Bifido species. So what we're really getting into with these type of studies is, being able to say, okay, your gut bacteria say that you're consuming too much protein. Okay, so as you know, as we all are aware, there are new diets on the market every year. And they just kind of add to, the existing. So you've got this spectrum of everything from, vegan to, carnivorous diets. And all the extremes. What if it's your gut bacteria that should be making the decision on what is the best diet for you? So I'll just put that out there. All right, we'll be looking at specific bacteria that produce butyrate and specific bacteria that produce propionate and specific bacteria that produce acetate. And then we discuss what the importance of these different short chain fatty acids are, as far as fuel source for enterocytes, this is, your gut cells in order to repair, scavenging ammonia, regulating the immune system, reducing oxidative stress, to, encouraging regulatory T cell differentiation in the gut, producing or promoting gluconeogenesis in the liver, supporting insulin sensitivity, improving gut hormone production, or, the fermentation of prebiotic fibers like inulin. And do it to this. Get out of the way. And absorb peptides. Okay. So we discussed the short chain fatty acids that we'll be reporting that are important for energy, for gut epithelial cells, like lipogenesis and gluconeogenesis, and be able to identify, that microbiota that alters colonic energy, host gene expression, and increasing inflammation. TMAO production. TMAO is very involved with, cardiac health. And that will be a key analyte. Secondary bile salt production. We've discussed in our other YouTube presentations about the importance of phase 2.5 detoxification. That has everything to do with bile flow and how bile is removing, detoxifying the body of toxins, and dysbiosis will unconjugate the bile, trying to carry out the toxins and cause a recirculation of toxicity. So, the more available, bile salts that we have, and the ability to reduce the dysbiosis is going to be a better detoxification. I'll point this out too. We get a lot of questions about, how do we reduce diarrhea, constipation, and bloating? And you see, it's directly tied into bile in many cases. So we're anticipating phase 2 to be released the end of July, and phase 3 the end of September or October. And phase 3 is going to, go even deeper. Urolithins, these have to do with aging and longevity, recycling, damaged mitochondria, and regeneration of new mitochondria. So energy production. And we'll also be talking about histamine versus indole production, some of these other indoxyl sulfate production. We'll get more into that when they get closer. So now some of the studies. All right, so this is a 35-year-old woman, has Hashimoto's disease, antibodies around 300, many food and environmental sensitivities, recent antibiotics, confirmed enteropathy and gastritis, via scope. She is taking Hist-AO by Zymogen and just started an elimination diet. So the first thing we look at is the alpha and the beta diversity. We can see we got some beta diversity issues. You see the alpha diversity is not good. We go by the colors here. And so when you say poor alpha diversity, we're saying the quantity of bacteria species is too low, and the beta diversity is showing signs of compositional dissimilarity. or the quality of the bacteria. And we've got some pathogens. We've got Clostridium difficile, C. diff, and Campylobacter. Here we're looking, again, that 30,000 foot view, just at the phylum. As we can see here, we've got a huge imbalance. You've got your Firmicutes ratio way too high, and you can see that visually when you look at the phylum. You know, you're supposed to have, you know, this nice mix, get some Firmicutes here, Bacteroides here, but here you can see you're just overpowered with the Firmicutes. The strobilome is showing very low, which means estrogen is being recirculated instead of being excreted. So you've got a level here you can compare to, and then you've got a visual. And then this is a diagram, again, showing you the relationship with the estrobilome. When it's high, you get intestinal reabsorption of conjugated estrogens. When it's low, fecal excretion of conjugated estrogens. So what we've got a situation of is the, it's kind of reverse, but we're looking at the reabsorption of the conjugated estrogens. The terminology is, we have to reverse it. So it's a high beta glucuronidase, low, Okay, these are the keystone strains, and this is what you want to look for when it's, not detected. Those are problems. So dysbiosis, defined as loss of beneficial organisms, excessive growth of potential harmful organisms, or loss of overall microbial diversity, excessive Firmicutes with low beneficial microbes can be aggravating to the immune system. Of course, you got Hashimoto's, so this is going to exacerbate that situation, and prone for estrogen dominance. So that would warrant further hormone tests. So what's recommended? A variety of plant fibers, resistant starches, anti-inflammatory fats, gluten-free, dairy-free, moderate protein intake, organic, support the liver and the bile with artichoke leaf extract. There just happens to be this great product called MegaGuard for that. Omega-3, and there's this great product called Omega that's designed for the gut, it's rich in EPA. Turmeric, curcumin, recondition the gut, and that's where you've got the spores. You add in some HU58 because you've got some pathogens in there, some IgG2000 to bind and neutralize the toxins from those pathogens, and Mega-Pre to increase that beta, that bacterial diversification in those keystone strains. Okay, just a little reminder again, why you want to heal the gut, to stop the leaking, to stop, this is really the source of autoimmune disease and inflammation and disrupting the immune system function. Right, another case study. We've got a 11-year-old female, neurological problems, behavior, sensory issues, sensitivity to light, constipation, limited diet, not taking any supplements. And this is just to remind us, this gut-brain connection. This child has neurological issues. So this is, showing, how microbial metabolites can feed directly to the brain, whether it's through, lipopolysaccharide or other endotoxins or exotoxins can damage the nervous system. And this is, just showing how stress and emotions involved in the limbic system and how you, again, can get these bacterial molecules affected directly into, the hippocampus, and affecting emotions. So we look at her initial index, and we can already see that there's an issue. The alpha diversity is horrible, and well, again, we're looking for extremes. And then we also see we've got some nasty pathogens on board. And here we've got some real out of whack firmicutes and some extreme proteobacteria. I mean, this is, yeah, horrible. Take the 30,000 foot view. We see, here's the amount of firmicutes. Firmicutes. But what we're really seeing here is how the proteobacteria is just blowing everything out of the water. And then just reminding you of, again, those bacterial endotoxins like lipopolysaccharide and how they really mess with so many systems of the body, mood, depression, cognitive decline, memory. I mean, so definitely can mess with behavior. Here we're looking at just a visual saying, there's only a few bacteria in balance that are helping her. There's a lot that are not there. So she's got very little resilience. And then out of the keystone strains, we're looking for, those, she's got very little helping her, and we got not detected, not detected, not detected. So she's really got, it doesn't have, the superheroes in there helping her at all. So the findings, we definitely want to go after the LPS and lower that to reduce the inflammation, stop the leaky gut, stop the leaky brain. Most likely the constipation is from the lipopolysaccharide shutting down the bowel motility. Very poor overall diversity. We have to eradicate the pathogens, changing the environment, maybe use some killing agents. At this point, once you've done the total gut restoration, if there's any still remaining, non-existent commensal organisms. So we really need those oligosaccharides on board. Just because they're not there doesn't mean they can't be revived. We've seen that before. So the recommendation is some high doses of the megaspore and the A258 and the Restore Flora to help with overgrowth of the pathogens, the IgG 2000 to help with the immune system modulation, omega-3 fatty acid to reduce the inflammation, perhaps some N-acetylcysteine or glutathione, support nutrient deficiencies, probably some methylation issues going on, get the omega prebiotic going for the oligosaccharide and help the keystone strains, support the liver, elimination phases of detoxification, remove any glyphosates that might be exacerbating the environment. All right, this is the final case study here. This is just to show a few points. We've got a married couple, almost identical microbiomes according to BiomeFX. Both are born vaginally, both breastfed, both born in cold climates, lived in warm climates, back to cold. They eat, exercise, lifestyle, same job. Male was exposed to secondhand smoke for 30 years, some moldy hay, and contracts some squamous cell carcinoma at the age of 60, and underwent 7 weeks of chemoradiation, but is currently PET scan negative. So when we compare these two microbiomes, I mean, we see, at the alpha diversity, got the beta diversity, and the resistomes. This is the individual that contracted the cancer on this side. Okay, and here we've got some comparisons just, to see that they're very similar. when we compare and contrast. And in some cases, some of the values were exactly the same, which just shows either microbiomes will sink because of environmental or other pheromonal type of things we don't understand. Again, showing similarities here. And the keystone strains. And when I said like this, here we got this Ruminococcus bromai and the value 87.56, 87.56. And these samples were sent in a month and a half apart from each other. So there was that lab didn't know that they were married. So this was, again, it does attest to, the lab accuracy. Okay. Again, very similar. The bifido got what, 78. Here we got 75. Okay. And ratios, look at this, exactly the same, 0.86, 0.86. Very interesting. Okay. But the difference is the individual that contracted the cancer, because of the chemoradiation, now has all of these pathogens on board. And there's some documentation to support these are very typical for chemotherapy. All right, so the conclusions. And let's go back to one slide here. We looked at lactobacillus. Over here, I can tell you this individual has no detectable lactobacillus, even though they have consumed yogurt almost every single day for 50 years. So we'll just point that out. So similar microbiomes may sink by environment, adaptation, or perhaps things we don't understand like pheromones. And then what I just pointed out, lifetime of eating yogurt does not increase lactobacillus. Chemotherapy predictably increases pathogenic burden. So, just to throw this out there for some questions, how long for repair? We're not sure. We'll have to get back to you on that. Of course, we would expect with this kind of damage for the chemoradiation, we're looking at probably 6 to 9 months. Yeah, and here we're just throwing it out there. Does a mate's microbiome empathize for the traumatized counterpart? We're dealing with, communication between these organisms that may extend beyond the quorum sensing of the gut. So, okay, so we'll end with that. Please text in your questions to these cell phone numbers, and we'll be getting back with you. And that's all for now. Take care.